Thanks for coming back from the coffee. We have another lovely set of talks coming for us. Our first speaker is going to stay plant-focused, Vincent Colot. And he's now a CNRS Director of Research at the École Normale Supérieure, just down the road from here. Up the road, sorry. I came the wrong way, that's why. He actually did his PhD at Cambridge in the UK and then had a bunch of different positions in France, and including at the Génoscope, and also spent some time at Cold Spring Harbor. He studies tomatoes and Arabidopsis and how other plants respond to transposable elements and how these transposable elements might contribute to adaptation and evolution and phenotypic variation, and does a bunch of epigenetic things as well. So we very much look forward to your talk.
Thank you so much for the very kind introduction. And thank you, Virginie, for giving me the opportunity to go downhill to present our work on transposable elements in plants. And indeed, there will be two aspects. One has to do with how transposons, through the mobilization, can generate or create mutations and phenotypic variation, but also how epigenetics sort of complicates matters and adds another layer of variation there that could be useful as well. So just so that everyone is on a par with me, let's start with what we know about transposable element sequences. So you know there are complex sequences to study in genomes because they are everywhere. In humans, we have at least 50% of our genome made up of such sequences, compared to about 2% of our genome coding for proteins. And in plants, in some plants, like in corn, for instance, you can have up to 85 to 90% of the genome made up of such sequences. But when we say transposable element sequences, what do we mean? Most of them are actually fossils, so they have long stopped to move. They moved in the ancient past, and typically, when you now look at mobile Ts within a genome, you have maybe 1 in 1,000 copies or even less that are actually mobile. So for instance, again, using humans, we know that only about 80 to 100 line elements, which are a specific class of elements, are mobile in our genome. And that's compared to hundreds of thousands of copies that are still present in the genome, but now highly degenerate because they have accumulated many mutations over time. Now, we know a lot about what transposons can do, or TE sequences can do at the macroevolutionary scale, from rewiring gene regulatory networks to helping placentation in mammals. So we have 7 occurrences of placentation in mammals, and all of these are due to the exaptation of one protein function that was encoded by an envelope of a retrovirus, an endogenous retrovirus. And we also have this beautiful system of VDJ recombination that generates the diversity of immunoglobulin genes in our lymphocytes. So all of these are actually driven by sequences that first were transposon sequences or transposon activities. Now, what we also know, of course, since the work of McClintock, who discovered transposons now over 60 or 80 years ago, is that they are powerful endogenous mutagens. And here I'm just going to highlight that through two emblematic examples, a wrinkle pea of Mendel and the Carbonaria form of the peppered moss. So in one case, we have something which is extremely deleterious with much less starch in the pea than you will need to germinate properly. And so this, we believe, is the most common situations where transposons, when they move into a gene, and here we're talking about mobile sequences, of course, when they move into a gene, they will typically kill that gene. But sometimes, they can actually do something else, which is to boost the expression of the gene, as in this case. And this turned out to be very important to let this peppered moss adapt to industrial revolution in Great Britain in the early 19th century. But beyond these functions, we also know that because transposons are mobile but also not mobile copies, are under strong epigenetic control in plants and vertebrates mediated by DNA methylation in large part, we know that they can exert their effects very differently as well. In addition to being mobilized, they can, when they sit in a place, depending on their epigenetic state, their DNA methylation status, they can be totally transparent to gene function, or they can alter this gene function dramatically. And here, this is a beautiful example in oil palm, where a transposon sits in an intron of a gene, which is involved in fruit development. And when the transposon is in the methylated form, which is a default state that you will find most TE sequences in the genome, somehow the gene is perfectly transcribed, the intron is spliced out, and basically you have normal fruit development. Whereas if by chance, and this chance is actually increased when you do in vitro propagation of oil palm, which is of course a big business in South Asia, in Indonesia, Malaysia, and so on and so forth, when now through propagation, in vitro propagation, you lose DNA methylation at this particular T sequence, now what happens is that basically RNA pol2, the polymerases that transcribe genes to make proteins, these get blocked upstream of the unmethylated T. So here what you have is one allele, but two different epiallelic states. And this epiallelic state, of course, is quite dramatic. So again, I maybe did not mention that properly, but transposons are characterized by when they mobilize to by the generation of large effect mutations. But you can also see that they can generate, even without being mobile, large effect epimutations. In this case, a totally sterile fruit. And what is fascinating is that this is not unique to plants. You can find it in mammals as well and in other vertebrates. But in plants, it seems that DNA methylation is very poorly reprogrammed at every generation. So once you lose by accident this methylation here, it will be propagated through sexual reproduction. So you can end up with almost Mendelian inheritance of such epivariants. So now the big question, and which is only beginning to be addressed in different experimental systems, is, what matters? Is it allelic variation that is brought about by transposon mobilization, or does this epiallelic version, which I just described to you, of importance as well in explaining phenotypic variation? And so to answer these two questions, and this is what my lab does, we turn to a model organism, which is Arabidopsis, for many reasons. It has a small genome, and when you want to do epigenetics, basically show that you can have variation without any sequence variation to start with, you have to be able to sequence your genome as often as required. And because it has a small genome size, we can sequence hundreds of individuals at very little cost. Despite having a small genome size, it has a very, very large repertoire of T families, each of a small size. So you have maybe 10 to 100 copies of a given T within a family, when in humans you have hundreds of thousands of copies. So this is actually an advantage for us, because we can now track individual T copies in the genome quite readily through genome sequencing. It is tolerant to dramatic loss of DNA methylation, which is a bit also counterintuitive. So you can remove DNA methylation of the T sequences and yet have plants which are viable and fertile. So you can do genetics with a very drastically hypomethylated genome and then try to understand how stable this hypomethylation can be across generations and what are the phenotypic consequences. Arabidopsis is a colonizer, so it has been now broadly distributed across the northern hemisphere, and we have a huge phenotypic variation. So we can now try to associate these differences in phenotypes with either T mobilization or T epivariation. And we do indeed have 1,000 genomes for which we have obtained whole genome sequencing data. This was a 1001 genome project, spearheaded, among others, by Magnus Nordborg, who is in the audience and will give a talk this afternoon. So we have an incredible resource here to explore this notion of T mobilization or T epivariation. And because we have methylome data and transcriptome data for many of these accessions, we can really put all of these together into one frame of study. Now, I want to give a tiny vignette about DNA methylation in plants. It's complicated. You can methylate your cytosines in a genome in any sequence context, CGs, just like in mammals, and humans in particular, with the same activity as has been described in mammals. But you have different methyltransferases that do what we call CHG and CHH methylation as well. And you have this pathway, which is fascinating, which is unique to plants, where two polymerases have evolved from RNA pol2 to now specifically transcribe repeat sequences and not to code any transcribe any proteins, but instead to just produce transcripts that will somehow feed back into chromatin and direct DNA methylation there. This is called RNA-dependent DNA methylation. And this is a kind of RNAi pathway where you have dicer activities, RDRT activities, sorry, to make a double-stranded RNA first, dicer, and then argonaute. And somehow this argonaute loaded with small RNAs will recognize a transcript produced by one of these polymerases, Pol5, which is a derivative of Pol2, which sits on chromatin with this long-hanging primary transcript. And this somehow then brings a de novo DNA methyltransferase to chromatin and will methylate the DNA. The cognate DNA, the one corresponding to this sequence here. So the first thing that we wanted to do, as I said, we are interested in finding out how transposons move, how often in nature, and what are the phenotypic consequences of that. So the first thing we did was to tap into this 1001 genome sequence resource and look for what we call tips for teeny transposon insertion polymorphisms. And basically there are two types, what we call the presence tip. So basically here this is your reference genome, where you have here the T that is actually fixed in the 1000 genomes and maybe at the species level as well. But you also have situations where a transposon is not present at this position in a reference genome sequence, but you do find it in some genomes, indicating that there is some transposition activity going on somewhere. And you can, of course, have the opposite situations where you have a transposon present in a reference sequence but absent from other genomes. So we use this information. This is Illumina short-read sequencing data. And look specifically for what we call discordant reads, where one read can be aligned to the reference genome sequence, but within a fixed size, because these fragments are typically 400 or 500 nucleotides, it does not map properly to the reference genome. It's because now this is corresponding to a T sequence, which has been brought in to this position in this genome or this genome, for instance. And we also have what we call split reads, which start with the reference genome sequence and then finish with a T sequence, which is absent from the reference sequence. So this is the extremities of these new insertions that are absent from the genome. These are the split reads. So we devised a bioinformatic pipeline that can tap into this Illumina sequencing data. And we also have a direct way of isolating T sequences from genomic DNA, and so we can experimentally validate what we observe through bioinformatic analysis. And just to cut a long story short, what we obtain is here just one example of the 5 chromosomes of Arabidopsis. This is a reference genome. What you have here are the reference genes. You can see that they are present at high density along the chromosome arms. And here you have the density of what we call Ts or T sequences, and are mostly concentrated in pericentromeric regions. But now, these are, and you don't ignore the different color codes, these correspond to different T families. This is an amalgamate of the 1,000 genomes that we analyzed, and we could detect over 23,000 transposon insertion polymorphisms. And what you can appreciate here is that most of these polymorphisms, compared to the reference genomes, they all, of course, have the same reference T sequences for the most part, but now, in addition, each genome has a few extra T sequences that we can detect, and they are along the chromosome arms. So this is already quite a striking observation that, these are nasty little sequences when they move around, and yet they love to go to genes. And it makes sense because this is open chromatin, and that's what we know in molecular terms, is that transposons go to where the chromatin is the most accessible, and the most accessible chromatin you can find is within genes. So here we have a system of endogenous mutagen that constantly creates mutation in genes. And so the concentration of T that we see in the reference genome sequence must be the result of purifying selection from insertion that occurred within chromosome arms close to genes. And indeed, so if we look now at the rate at which these transposon insertions arise, we just considered basically pairs of strains which are very similar to each other, and we simply ask what is the number of transposon insertion polymorphisms that distinguish one and the other of the two pairs of strains. And so when we do that for very, very closely related pairs that differ by less than 300 SNPs, we find that transposition accumulation is just 1/3 of the rate at which you accumulate SNPs. So we know that SNPs are accumulated because of errors during DNA replication or during DNA repair. But what we find now is that in nature, transposition is almost as frequent as the accumulation of replication errors in your genome. And given that these correspond to insertions near or within genes, this should have absolutely horrendous effects on genomes. And indeed, when we look now at how these TIPs distribute in terms of population frequencies, unlike SNPs, which many of them can reach very high frequency in populations, TIPs tend to be very low frequency variants, suggesting that they are never sort of sustained for a long time because preferring selection will be there to eliminate them. And that's exactly what we observe when we consider the entire set of data that we have for the 1001 genomes. This is a rate of TIPs accumulation that we expected based on the closely related strains. But you can see we only recover between any two strains, the most divergent one, 730 TIPs, so 0.2% of the predicted accumulation that we will have had if these things could be accumulating neutrally, like most SNPs. So there is very, very strong counter-selection for these T insertions that do occur nonetheless in the genome. So based on this analysis, what we then did was say, okay, let's just focus on, we're interested in T mobilization. This has never been characterized to that extent in natural settings. So let's consider the TIPs that are the lowest frequency and for which we have some good evidence that these correspond to very young insertions, maybe 1,000 years ago, 10,000 years ago, but very young compared to most of the T sequences that you will find in the genome. And what you appreciate immediately is that there is definitely quite a wide variation in the number of TIPs, of recent TIPs, so basically of mobilization activity between genomes in nature. And so we use this trait, number of recent insertion per genome, to do a GWAS, a genome-wide association study, to see if there are some genetic determinants that will modulate the rate at which T mobilization occurs in nature. And sure enough, when we did that, we came up with one very strong peak of association, which maps at one locus, which is called NRP1. NRP1 is the largest subunit of this POL5 derivative I talked about, which is responsible for RNA-dependent DNA methylation. So we have natural variation for this particular gene in nature, and actually Magnus, some years ago, identified it as a key component in variation in CHH methylation, which is, it's no big deal. CHH methylation is typically over a T 10 to 15% in level. So here you have a slight reduction, but now we have a phenotype associated with this subtle reduction in CHH methylation, which is increased transposition. When you have this non-reference allele, you have much higher transposition rates than when you carry the reference allele. And then we went a bit further and tried and explored whether there could be also G by E interactions. And sure enough, we found beautiful G by E interactions with this particular variant as well, not with the reference allele. So here what we believe we have is an indication that NRP prime, which segregates in nature in very specific environments, is actually an environmentally conditioned mutator allele. So it will provide more potential for large effect mutations in environments where it might matter to have, to find new variants, because remember, this Arabidopsis is a colonizing species, so you won't have where at the edge of the niche, you will need to come up with new solutions because your population size will be very minimal and your sonic genetic variation will be very reduced. So de novo mutations might be very important under such situations. And here, just I realize that I'm getting a bit short with time, so I just want to highlight one particular region that we found where one gene where we had a surfeit of T insertion polymorphism. So basically, at this particular locus, I will tell you what it is in a minute, we found 16 occurrences of T insertion alleles, each independent from each other. But look at that. They are all within just one intron, nowhere else. So the gene is still functional. And this gene is actually one of the major determinants of flowering time in Arabidopsis. So what we have here, and we know that this particular intron is the regulatory intron that modulates expression of FLC. And this modulation is essential for what we call vernalization, the need for some of these strains of Arabidopsis to overwinter so that they can flower at the next spring. All of these insertions have one effect, which is to reduce the expression of FLC, which is a repressor of flowering. And so when you reduce the expression of FLC, what you do is you reduce now the flowering time. So you get an edge compared to your neighbor. You maybe flower 15 days earlier than your immediate neighbor that has exactly the same haplotype, but without the insertion. This is the carriers and the non-carriers for these insertions. Every time that we have it, we have a tendency to flower much earlier. So we believe that this is a beautiful example of repeated local adaptation using Ts as generators of these large effect mutations. And sure enough, what we don't have is T insertions with these essential genes. We never recover them in nature. They can't be tolerated. They are too dramatic. So we have the number of genes for which we have never recovered in insertion in nature is pretty high. Actually, probably 2/3 of genes have never been visited, do not seem to be visited by transposon in nature. Well, they might be visited, but they are immediately counter-selected, and among these, the ones that are associated with essential functions predominate. Okay, so enough about T mobilization. So remember, very few Ts are mobile in a genome, and yet we see the dramatic consequences that these T can have on generating large effect mutations in natural settings. Now let's move to epivariation, much more complicated to study because epivariation in nature will, of course, co-segregate with sequence polymorphism, so you need to find out, to tease apart the two sources of variation in relation to phenotypes. So how did we go about that? Well, the first thing we did was to go back to the lab and take advantage of one particular mutant that was described a long time ago, DDM1, for decrease in demethylation 1. When you mutate DDM1, you basically completely dissolve this possibility to methylate T sequences. And this is because DDM1 is what we call a chromatin remodeler. It sort of moves around nucleosomes, and that allows the methyltransferases to gain access to DNA. So when you remove DDM1, the methyltransferases have a hard time to access repeated sequences, so they cannot maintain the demethylation there. So we used this property of DDM1 to do a very simple genetic experiment. Start with 2 strains that are from the reference strain, Columbia, but one carries now the DDM1 mutation. So in this strain, in this mutant, all the Ts are severely hypomethylated compared to the wild type. Genes are the same, but the Ts are now severely hypomethylated compared to the wild type. So we made an F1, we backcrossed it to the wild type, and we recovered in a backcross progeny the 50% of individuals which are now homozygous for the DDM1 locus. So everything is back to normal in terms of capacity to methylate your genome. But the big difference is that some of their chromosome intervals will have been inherited from the DDM1 parent. And so we generated from these individuals what we call epigenetic recombinant inbred lines. So basically what we do is we recombine epigenetic state, methylated and methylated. Same genome sequence. The only thing that can recombine is these two methylation states. And what we found is that a fraction of sequences, T sequences, can be propagated very stably in hypomethylated states for many generations. This is only 8 generations here. We push our lines to 20 generations. They are still hypomethylated. So this is, again, the illustration that at least in plants, you could exist in 2 very stable, different epigenetic states, and they will be transmitted according to Mendel's laws. But at the other extreme, we do have sequences for which we remove DNA methylation through the DDM1 mutation, which now regain it with extremely high efficiency. And we have a complete continuum of situations between fully stably inherited, and fully reverting. And we realized, so this was published a long time ago, but we reevaluated that with new technologies, and now we realize that what matters to distinguish between these possibilities is 23 minutes and 38 seconds. Okay. It's copy number. So basically, these sequences do not exist in isolation. They are T sequences. So they have all of their related colleagues, copies, present elsewhere. And the difference in stability that we find between these has to do with the number of related T copies that come from the wild type. The more related copies come from the wild type, the more capacities you have to regain methylation, because only the wild type actually still has a possibility to produce the small RNAs that will instruct gene methylation. So this is, in essence, what we found, is that these sequences can be heavily methylated. They will lose methylation in DDM1, but some of them will be fed back to the methylated state because trans RNA coming from wild-type copies segregating elsewhere in the genome. So this is all experimental, no sequence variation. This is truly epigenetic. Now we can move to nature. And when we move to nature, what we find is that we had about 7,000 of these, what we call T differentially methylated regions, so TDMRs, 6,000 of them, no variation in nature. They are systematically heavily methylated. But for 1,000, we definitely do see epi variation in nature. And what is more remarkable is that this epi variation somehow follows the rule that we established in the lab. We find more epi variation in nature for sequences that in the lab we can detect as being stably transmittable in the hypomethylated state. And very few that in the lab tend to revert at high frequency because they are targeted by small RNAs coming from many other copies in the genome. And even their frequencies follow this. So basically, we believe that in nature, we have also the same system playing out, not through DDM1, but nonetheless. We find that the Ts that are epivariable in nature tend to be close to genes, much closer than at random. And they even are closer when they are at high frequency, suggesting that basically epivariation at T sequences in nature might be associated with the fact that they are close to genes. So we did a GWAS based on this property, indicating here the number of hypomethylated T DMRs you find in a genome. And we, again, found just one peak of association. And it maps to a single gene, which is totally functional. We worked that out through AlphaFold prediction, but which is overexpressed in a variant form compared to the reference allele. And this variant form is associated with a higher number of hypomethylated sequences in the genome. What is ELF8? It is a key component of the transcriptional machinery responsible for the transcription of genes by POL2. And basically, ELF8 antagonizes the function of RDDM. So here we have a mechanistic explanation as how the two can play out. So basically, in nature, if a T sequence is near a gene, it will have a tendency to lose its methylation because the gene will be expressed. And now I will actually finish here with just one study, which is comparing for, so here what we are dealing with are T sequences that are present in the reference genome sequence. Most of them are fossils, have been there for ages, but not all of them are fixed. So you can have situations where even for reference T sequences, they are present in the reference genome, but absent from some genomes. And they could be present in two different states. In the reference genome, they are fully methylated. That's how we started with. But they could exist in the apomethylated state. So we took all the cases where we had these three situations, epiallelic variation as well as allelic variation. And in blue is the results of comparing gene expression, nearby gene expression when you have allelic variation or when you have epiallelic variation. And I think you can appreciate that wherever you look close to genes, the effect of this allelic variation is much less than the effect that you have of the epiallelic variations, which are in green. So the blue is always weaker than in green. It makes sense. These are reference T sequences. They have already been checked by natural selection just for the property of inserting next to a gene. If they had been deleterious, they would have been eliminated. So they have very little effect to start with. And yet, what we can reveal now through epivariation, that they bring a new level of phenotypic variation on gene expression. And this is heritable as well. We have many examples where we could follow in what we call recombinant inbred lines these two different states and show that are stably inherited, independent of sequence variants. So to summarize what I showed you today, is that T mobilization is a major source of large effect mutations in nature, but that as a result, these are extremely rapidly eliminated by natural selection. That this activity is nonetheless modulated in nature through a key genetic modifier that seems to play out in an environmental dependent fashion. So we have this environmentally conditioned mutator allele, perhaps. That T mobilization can contribute decisively to local adaptation. This is an example of 16 independent insertions that shift flowering time by at least 15 days in many places in the world. But that also, there is a potential for epivariation, even for these old T sequences. And this potential is indeed, in part, found in nature to be happening. And this could definitely contribute to phenotypic adaptation, as well as, the large effect mutation that we talked about initially. The big difference is that here, we are dealing with old sequences that will be present at much higher frequency in a genome. And so we could, again, imagine that they can adapt locally little subtle variation in gene expression levels that will be, again, adaptive. So both mobilization and epivariation at TEs could contribute not only to phenotypic variation, but perhaps to adaptation. And these are the people I need to thank, mostly Pierre Baduel and Luna de Oliveira, two bioinformaticians, and Leona Quadrana, who was a postdoc in the lab and now has his own group near Paris in Saclay. Thank you.